TK, the science guy.
TK, the science guy.
TK, the science guy.
Apes rule.
TK, the science guy.
Wind power, I'm a big fan.
Time.
TK, the science guy.
TK, the science guy.
Hey CW Bio, Mr. Kennedy here. Today I've got all kinds of equipment laid out in front of me so we can investigate photosynthesis and cellular respiration and kind of see how one plays into the other. What I'd like you all to do is to go to our class webpage and find this handout, Photosynthesis and Cellular Respiration. Make a page for it in your notebook and come back when you're ready to get started. I'll see you in a few minutes. Welcome back. Let me take you through how all of the things out in front of me here are going to be used to investigate photosynthesis and cellular respiration. I've got 1, 2, 3, 4 test tubes. I've got a little bit of water, an indicator called bromomethyl blue, some baking soda, good old-fashioned table sugar, and our test subjects, yeast, and a plant called Elodea. Looks yummy. So what I'm going to do is I'm going to follow the instructions in the lab, add these things to the test tubes, and, get the experiment going. I'm going to take all 4 of my test tubes and I'm going to put water in them. I'm going to put bromomethyl blue in them. I'm going to put some yeast in them. I'm going to put some Elodea in them. And a couple of these tubes are going to be put in bright sunlight, and a couple of the tubes are going to be put in the dark. And hopefully, if everything goes the way we want it, we're going to get to see them change color based on photosynthesis and cellular respiration. It's the byproducts of photosynthesis and cellular respiration that are going to interact with the bromomethyl blue to cause a color change inside of all of our test tubes. So, I'm off to set everything up and get it running. I'll see you in a minute.
Here we go.
1 hour later.
Whew, that was a lot of work. Well, everything's all set up, so now I have 4 test tubes, and inside each, they've got a little bit of plain old table sugar, some yeast, a water plant called Elodea, or Anacharis, a blue indicator called bromomethyl blue, and a little bit of baking soda. 2 of these test tubes are going to be put in the dark, and 2 of these test tubes are going to be put in bright light, like outside. And we're going to wait to see what happens. Right now, they're blue, so maybe we'll get to see a color change. Let's see. I'll see you in about an hour.
1 hour later.
I'm back. It's been, about an hour, and whoa, there was a color change. Can you believe it? The test tubes that I left in the dark turned yellow, and the test tubes that I left out in the sun, they stayed blue. That's weird. Like, how come these reacted, but those didn't? Or maybe did they both react? Am I missing something? You tell me. Let's think about photosynthesis and cellular respiration. What does each one make as an end product? If you remember what we learned in class, photosynthesis takes in carbon dioxide and then it releases oxygen as a waste gas. Cellular respiration, it takes in oxygen and it releases carbon dioxide as a waste gas. Now, Little known fact is that when carbon dioxide mixes with water, it forms carbonic acid. What? Yeah, it forms an acid. So as we're looking at our test tubes here, bromomethyl blue also changes color in the presence of an acid. It turns yellow when an acid is present. That might tell you something about what's going on over here versus over here. Now, Mr. Kennedy doesn't want to just give away all the answers, so I want you to think. Why would these tubes turn yellow in the dark? Why would these tubes stay blue in the sun? Interesting question. Well, did you figure it out? I think I know what happened. But I'm going to wait until I read your lab reports in order to find out if you know what happened.
TK the Science Guy.
TK the Science Guy.
TK the Science Guy.
Apes rule.
TK the Science Guy.
Wind power. I'm a big fan. Time. TK the Science Guy.
TK the Science Guy.